안녕하세요 and 감사합니다. My name is Alastair King. I'm the head of biology at Eurofins Discovery, and I'm going to speak to you today on the use and power of using cellular assays with database mining in oncology drug discovery. These are the topics that we'll be covering. We'll talk about the classical anti-cancer drug evaluation process and the way that this is carried out in discovery. We'll identify we'll talk about the way in which we can identify the most susceptible cancer types for a particular drug in development. We'll talk about also how we can identify predictive genomic biomarkers for both sensitivity and resistance that can be useful for potentially stratifying patient populations in clinical testing. We'll also look at broader activity or consequences of a drug and what it does outside of the tumor. This includes a quick evaluation of how we can look at the tumor microenvironment, and especially from the context of immuno-oncology agents. We'll also talk about the way in which those drugs can be evaluated using a complex phenotypic in vitro cell system, and that cell system can also be used to look at broader effects, in particular predicting safety of drugs and potential toxicity profiles. And then we'll wrap up with a summary. So this is an overview of the typical drug discovery pathway. And it outlines the different activities from identification of a target through identification of compounds or hits from a screening process, through establishing activity, optimizing that activity, and ultimately selecting a candidate for evaluation in clinical testing. Obviously, there are a lot of things that need to be done to get to a candidate drug for clinical testing. And what we'll talk about within the context of this webinar are some of the approaches that can be used using cellular assays, not only to look at efficacy, but also safety aspects in drug development. So to start with then, the first cellular assay that one typically uses in really any drug discovery program is an evaluation of what is called mechanistic activity. In some cases, this is called target engagement activity, but simply put, it is a cellular assay that is designed to show the effect of the test agent at the target with a readout that is a direct consequence of activity at that target. So what I'm showing here is a simple measurement of phosphohistone H3, so histone H3 phosphorylated, in this case at serine 10, as a marker for aurora kinase activity. So this study was conducted in A549 non-small cell lung cancer cells. They were arrested with nicotazole to synchronize the cells in a particular phase of the cell cycle, and then treated with various aurora kinase inhibitors. So you can see from the graph shown here that we can look at this particular inhibitor and measure its potency. Now, this particular inhibitor is a pan-aurora inhibitor. It inhibits auroras A, B, and C. If we look at this particular graph, we also have a pan-aurora inhibitor, and we can show a different potency in the same assay. So to start with here, we can rank order compounds by their potency. If we look at this particular graph in the top right, this is an aurora A and B inhibitor, so it lacks the C inhibition component. And again, we can measure its potency. We can also look at an aurora A kinase inhibitor that is selective for aurora A. We can look for inhibitors that are selective for aurora B. And in each case, we can measure the potency of the particular inhibitor in this assay. Now, the important thing to remember here is that this is a target engagement assay or a mechanistic assay. And so it shows the activity of the agent, of the drug at the target, but it does not give us any indication of the functional activity of that drug in terms of inhibition of tumor cell viability or proliferation, which of course is the ultimate goal for any oncology drug. So we then typically turn to an assay that looks at the effect of drugs from a functional perspective. So what I'm showing here are a panel of different assay components with different cell lines. And this is using vemurafenib, a targeted BRAF kinase inhibitor, when incubated with a number of different cell lines. So the column on the left, the three graphs on the left, show the effect in C32, which is a cell line, a human tumor cell line that contains the BRAF V600E activating mutation. And the graph in the top left shows the effect on proliferation. So what you can see here is a dose-dependent inhibition of proliferation. And we would expect this of vemurafenib because it is a targeted BRAF inhibitor, and it was designed to inhibit the growth and viability of human tumor cells that express mutated activated BRAF. Now the graph below it on the left-hand side is taken from the same experiment, and this is using a multiplexed phenotypic assay where we use DAPI nuclear staining to measure the effect on proliferation, and we can look at the effect on induction of apoptosis. This is done by measuring levels of cleaved caspase-3, and in this particular graph you can see that there is a dose-dependent increase in caspase-3 cleavage that is concurrent with the effect on proliferation. Now what I will quickly mention are the several data points on the right-hand side of that graph that are open circles. These are open circles, and you'll see that the curve does not go through those points because we do not count them in the analysis. The reason for that is because at those concentrations of the test drug, there are so few cells left, and you can see that in the proliferation graph above, with which to normalize the signal. So even small changes in the few cells that are left can provide a very large change in the apparent signal. So we do not use those. But nonetheless, you can still see an increase in apoptosis there with this particular drug in this cell line. Now below that, in the bottom left-hand corner, is another part of the multiplexed evaluation where we are looking at the effect of the drug on the cell cycle. And we're doing this by measuring phosphohistone H3, this time looking at serine 28, and a decrease in the signal relative to the vehicle control, which is set at a signal of 1, indicates a G1S arrest. An increase would indicate a G2M arrest. So we can see here that vemurafenib in C32 cells is acting through what looks like a G1S arrest. Now if we go to the middle column, we're looking at NCI-H69, which is a human tumor cell line that does not contain a BRAF activating mutation, but it does overexpress the oncogene c-KIT. Interestingly, you can see an activity there in the top graph for proliferation inhibition using vemurafenib. And then in the middle graph, there is a modest but a noticeable increase in apoptosis. And then at the bottom may be a very slight change towards G2M arrest. So clearly in this particular cell line, even though it does not contain the BRAF activating mutation, it contains an overexpressed oncogenic driver, and in fact c-KIT can potentially signal preferentially through BRAF. And that might explain why this cell line has some degree of sensitivity to vemurafenib. Now on the right-hand side, in the SAOS2 osteosarcoma cell line, this is also a c-KIT overexpressing cell line, so again, it does not contain the BRAF V600E mutation. And what you see again is a dose-dependent decrease in proliferation. However, in this case, you will notice that the Y-min of that curve is not at 0. So we either have an incomplete response here or a cytostatic response. Now this would suggest a different genetic makeup and thus a different response of this cell line to vemurafenib as compared with the c-KIT overexpressing cell line NCI-H69. If we look at the graph below that looks again at the induction of apoptosis, we see a very strong induction of apoptosis. So what this would tell us then is that the proliferation graph above is indicative of a partial response, and during that partial response, a subset of the cells in the culture are actually undergoing cell death. And again, because that induction of apoptosis is concurrent with the decrease in proliferation. We also see what looks to be a G1S arrest in the cells as well. So what we can see here are different effects in different human tumor cell lines by the same drug, and in certain cases we see activity of that drug in cell lines that we perhaps wouldn't otherwise expect it to have activity. So along this similar line, if we now look at the effect of trastuzumab, a very selective HER2 and monoclonal antibody, and we look in a variety of different breast cancer cell lines, we again see different responses. You'll note in yellow, the line at the top there for BT549 shows no response at all for this particular cell line. Whereas in the blue, the turquoise blue line, we see a dose-dependent decrease in proliferation for BT474. And then in the green, for SKBR3, we see a similar dose-dependent decrease in proliferation, but with a different potency. Now, what you'll also notice is that there is an effect on the T47D cell line in purple. It shows a different dose-response curve, but nonetheless, there is an effect there. What I will say, though, is in this case, all of these effects show a partial response. And in fact, trastuzumab works not only by inhibiting proliferation at the level of HER2 inhibition, it also induces antibody-dependent cytotoxic responses, ADCC, and that contributes to its therapeutic activity. Now, if we look at these cell lines from the perspective of whether they are HER2 positive or HER2 negative, to start with BT549, the cell line shown in yellow, it is HER2 negative, and it does not respond to trastuzumab. This we would expect, and this is why trastuzumab was developed to target HER2 positive cancers. BT474 and SKBR3 also show activity, and they are also HER2 positive. So again, we are seeing a response here as we would expect for this monoclonal antibody. But T47D is interesting because T47D is negative for HER2, and yet it actually responds very potently, very sensitively to trastuzumab. So what is happening here in this cell line that makes it so sensitive to trastuzumab when it's not HER2 positive? So if we pause that thought for a moment and think about what we can do to identify potential genomic features that might dispose a cell line to sensitivity to a drug, if we look at a large collection of cell lines, and we're looking here at upwards of 240 different human tumor cell lines, and you can see they cover a wide range of different tissue indications as shown by the key on the right. If you look at trastuzumab and you plot its log EC50 against the cell lines, each vertical line in the graph represents a different cell line, you see that the vast majority of the human tumor cell lines here are insensitive to trastuzumab, again, as we would probably expect. There is a cluster of cell lines at the left-hand side that show sensitivity to different degrees to trastuzumab, and you will see the annotation here. There are several breast cancer cell lines there that are HER2 positive, and as we would expect, respond to trastuzumab. So we can see visually here that there may be some association of HER2 expression with trastuzumab sensitivity. In contrast to this, if we look at doxorubicin, which is shown below, doxorubicin is a broad-spectrum cytotoxic agent. So you can see the dose-response curve, or the waterfall plot here, is a different shape. Every cell line shows some degree of sensitivity, albeit to a different level. And this is very typical of broad-spectrum cytotoxic drugs because they show activity in many, many cell lines across different tissue types, and they show a different waterfall plot when rank ordering the cell lines by their relative sensitivity. So what we can potentially do then is with any kind of plot like this, we can mark out cell lines that are in what we would call a sensitive group. So for trastuzumab, you can see that very easily. That would be the cluster of cell lines on the left-hand side of the top graph. We can also mark out cell lines that are the least sensitive. Now for trastuzumab, that would be all of the insensitive cell lines. But for something like doxorubicin, it may be, say, the 40 cell lines on the right-hand side of the bottom graph. And we can set cutoff limits there that delineate sensitivity versus resistance. Now we have tremendous genetic information available on the vast majority of cell lines in the collection. These data were taken from the Broad CCLE database in Boston and the Cosmic Sanger database in London. And we have actually combined those two data sets to form one proprietary database. And we can interrogate that across more than 274,000 different genomic features and across over 19,000 different genes. And we can statistically ask the question, are there any mutations or differential expression patterns for any gene that correlate with the cell lines that we are saying are the most sensitive and the cell lines that we are saying are the most resistant? So when we do that then, we can look at the various cell lines in such a study. And just focusing for a second on the graph at the top left, and this is looking at vemurafenib again, what you see are a number of tissue types, and they are shown towards the top of that graph, that are really not that sensitive towards vemurafenib. But then as you go to the left, you gradually see cell lines with greater sensitivity, and ultimately, in the mustard-colored box and whisker plot on the left-hand side, are the melanoma cell lines. Now these show the greatest sensitivity, and as we might expect, because the BRAF V600E mutation is quite prevalent within the melanoma tumor type. So this pattern that we see here mirrors what we see and know for vemurafenib, but can we statistically show biomarkers and identify biomarkers from this study that make sense of how vemurafenib works? Well, if we conduct orthogonal statistical assays, and the reason we use two different statistical tests is that each statistical test has its own mathematical bias, and we do not want mathematical bias to influence the observations and the data that we generate from this study. We want the biology to really direct the observations we see. When we conduct statistical tests to work out p-values for association with sensitivity or resistance in those two groups that we'd outlined previously, and then we plot the odds ratio against the effect, you can very clearly see in the plot at the top right that BRAF mutation or amplification of function correlates very strongly with sensitivity, while genes such as KRAS mutation, P53 mutation, or FBN2 mutation correlate very strongly with resistance. And these make sense from the context of BRAF inhibition and what we know about BRAF inhibition and how BRAF mutations drive certain cancers. If we also look at differential gene expression, we can identify genes that also are overexpressed in sensitive and resistant groups. So what you can see here then is that we have identified BRAF mutation or amplification as being a key genomic biomarker for sensitivity. As I say, this is what we would expect of vemurafenib, knowing how it works. But for a new drug, and especially if that new drug is targeting a relatively novel molecular target, the genetic determinants of what make that tumor sensitive to that drug may not be known. So this kind of analysis can use database mining to identify potential markers, and those markers can then be used to stratify patient populations in a clinical trial, and thus potentially increase the likelihood that that drug will show a clinical effect when in that trial. Now, coming back to trastuzumab and that question we had around the T47D cell line, how is it that a selective HER2 monoclonal antibody could show activity in a cell line that we know is HER2 negative? Well, if we do an oncopanel study like this using trastuzumab, and we look at the mutations, in the table at the left, you can see that ERB2, or the gene for HER2, when amplified, is the most significant gene found there in the mutation analysis. Again, this is what we would expect for trastuzumab. Excuse me. When we look at differential gene expression, and that is shown in the table on the right, we also see ERB2 overexpression as correlating with sensitivity to trastuzumab, and that's shown in the green. There are two entries here because there are two different probes that are used. But what you'll also notice is ERB4 is there. Now, ERB4 is also a member of the EGF receptor family, as is ERB2. But ERB4 is correlating here with sensitivity to trastuzumab. So the question is then, why is this? Why do we see sensitivity for cell lines that overexpress ERB4 when trastuzumab is a very selective HER2 antibody. So this potentially has identified a biomarker for sensitivity here that would suggest that anti-HER2 antibodies such as trastuzumab may have activity in human tumor cell lines that overexpress ERB4. The key question then is, why is this happening? If we look at the biology around HER2 and ERB4, HER2, when it's typically overexpressed, it does not have an extracellular ligand binding domain. So when it's overexpressed, it forces dimerization of the receptor, and that induces signaling in the cells. And this is what we associate with HER2-positive tumors. If we look at ERB4, ERB4 is interesting. ERB4 does have an extracellular ligand binding domain. And when the ligand binds, the receptor dimerizes, and that induces signaling inside the cells. This can also be manifested by ERB4 overexpression in certain circumstances. And in fact, ERB4 overexpression is known to be a tumor suppressor. However, when ERB4 is overexpressed and HER2 is present, not necessarily overexpressed itself, ERB4 in an oncogenic situation can heterodimerize with HER2. So what you then have here is oncogenic signaling, and that is shown in the graphic at the bottom left. ERB4 homodimerization acts as a tumor suppressor, but in circumstances where ERB4 heterodimerizes either with EGF receptor or ERB2 or HER2, this acts as an oncoprotein. So in cell lines that overexpress ERB4, if there is any HER2 present, it doesn't have to be overexpressed, but it is present. And even HER2 negative cell lines do express low levels of HER2. In this particular case, you can then see why trastuzumab would have an effect on T47D. If you prevent that heterodimerization, you do not get that oncogenic signaling. And in fact, if ERB4 is overexpressed and it cannot heterodimerize, it is very likely it will homodimerize and then also act as a tumor suppressor. So with this in mind then, you can actually rationalize why trastuzumab showed activity in the T47D cell line, and where we've actually identified a predictive genomic biomarker that identifies tumor types here that may be sensitive to a drug where they were not necessarily thought to be so. So this can potentially open up new possibilities for a drug in terms of targeting other tumors where you wouldn't normally expect it to have activity. Now, if we turn for a second to the other part of drug discovery, we've talked a little bit about efficacy here, but if we think about what can we do to reduce attrition. So I'm going to introduce to you and explain the use of a translational phenotypic platform called BioMap. BioMap is, as I say, a phenotypic platform that makes extensive use of human primary cells and co-cultures. The reason human primary cells are used is because they are very good at modeling particular tissue biologies and diseases. They haven't effectively forgot where they come from, and they act still like the source tissue from which they were derived. We have a number of models or systems of different aspects of human tissue biology and disease, and we currently have about 50 different models. They are comprised of human primary cell types in co-culture, and we measure a variety of biomarkers from those models when stimulated with physiologically meaningful stimuli that model the biology or disease being examined. Those biomarkers are clinically relevant and have been known to be modulated in clinical settings. Now, what makes our platform, the BioMap platform, particularly powerful is the huge reference database we have. We have profiled over 4,500 different approved drugs, clinical entities, things that have been into the clinic and in some cases failed, because those two can yield very valuable information that can be used in a platform like this. Small molecules, biologics, lots of different therapeutic modalities, a wide range of different molecular targets and phenotypes. Now, what we can then do is we can use that database when we test a new drug against BioMap, in particular in an offering called Diversity Plus, which I'll explain on the next slide. We can look for similarities with known profiles. And from that, we can identify things like mechanism of action. A lot of groups these days are looking at phenotypic screening, where a desired phenotypic endpoint is achieved, but the mechanism is not clear. So using Biomap by similarity searching, we can potentially identify that mechanism of action from this phenotypic cell in vitro cellular platform. Similarly, we can also look for potential toxicity signatures, again by aligning features in a profile with known toxicity profile signatures that come from the evaluation of drugs where we do know their toxicities. We can look at the effect of drugs from a combination analysis perspective, and we can also benchmark new potential drugs or lead compounds, candidates, with existing standard of care agents to show potential differentiating features. This is a very quick overview of the Biomap Diversity Plus platform. It comprises 12 different models, each model modeling a different aspect of human tissue biology or disease. Very loosely, I've grouped them here to show the fact that it covers broad biology, and some tissues model different aspects of biology. For example, in the top left in the blue outline, we have models that look at vascular biology. In the yellow outline, we have models that look at fibrosis. In green, those that look at respiratory biology. And in red, those that look at immune biology. So across this panel of 12 cell lines, we have a diversity of different biologies modeled. We can also look at things like cell proliferation and anti-proliferative effects, or even cytotoxic effects. Now, we evaluate a new drug against this platform, and we measure 148 different protein biomarker readouts across the 12 systems. And this gives us what we call a molecular phenotypic fingerprint. This is an example of such a fingerprint where the 12 systems are shown across the top, grouped by the different biologies that they model. What I will first describe is the gray line. If you look at the datum line that goes from left to right, there is a gray shading behind it. That represents the 95% significance envelope from historical data for vehicle testing. So you can see for different biomarkers and different systems, different models, we see different levels of variance of the biomarker. So this represents effectively the vehicle control or the isotype control in certain cases. We typically test an agent at 4 different concentrations. And what you can see here for this particular drug are that there are some very nice responses in several biomarkers across this profile. You see some nice dose-responsive markers. What I should also mention is that the Y scale is a log scale. So the differences between these data points is actually larger than it appears here. So this generates what I call that molecular phenotypic fingerprint. And it is that fingerprint that we can go back to the database and say, does anything look like this? As you will also note in this particular profile, in the BT and the SAG system, there are 2 indications of an anti-proliferative activity. So again, providing more information on how this particular drug works. Now, as I'd mentioned, we can profile a novel agent in Biomap Diversity Plus. We can analyze the profile to identify new biomarkers, and they may be useful from a clinical perspective. We can benchmark with either known agents or looking at 2 potential lead candidates. We can do similarity matching, and this gets to the identification of mechanism of action. And so there are several ways in which we can do that by simple statistical database matches. We can look at mechanisms from a heat map perspective and even similarity networks. Now I'm just going to take a quick step to one side to take a quick look at some of the aspects of Biomap where we can model the tumor microenvironment. This is particularly important from an immuno-oncology perspective. And to cover this very briefly, we have 4 different models, each in 2 pairs. One pair look at the stromal microenvironment, and they are conducted by mixing a human tumor cell line, the example here shown being HT29, with immune cells, and we use PBMCs in this case, peripheral blood mononuclear cells, and fibroblasts. So this models the stromal matrix. We have a pair of models, and again the example shown here is for HT29, that model the vascular component of the microenvironment. In this particular case, we co-incubate the tumor cell line with PBMCs, But in place of fibroblasts, we use human umbilical vein endothelial cells. So we have this pair of particular systems for HT29. We also have an analogous pair using NCI-H1299, a non-small cell lung cancer cell line. So we mix the cells together in co-culture. We stimulate them with appropriate ligands to model the biology being evaluated. We treat with the test agents, and then we measure a set of biomarkers in the model that are indicative of the biology being evaluated. Now, what we can show really with the way that these tumor microenvironment models are set up is shown here. Just going from left to right, human dermal fibroblasts alone, when they're stimulated with an appropriate stimulus, so that, for example, a T-cell receptor agonist, they do not produce interferon gamma. PBMCs, on the other hand, do. So you see a nice induction of interferon gamma production. When you mix PBMCs with fibroblasts, that production of interferon gamma can be accentuated even further. Now, HT29, the tumor cell line, a colorectal tumor cell line here, does not show the induction of interferon gamma, either when it's alone or incubated with human dermal fibroblasts. When in the presence of PBMCs, there is an induction of interferon gamma production, and you see that in the full model, the STRO HT29 at the bottom right, where all three cell types are co-cultured together. But what you'll notice is there's a tremendous suppression by the HT29 tumor cells of the production of interferon gamma. And this is a marker effectively for the suppression of immune surveillance that allows tumor cells to evade that immune surveillance, and is in fact the reason why there has been tremendous success in evaluation in the world of immuno-oncology. If we look at the effect of an anti-PD-1 drug, and in this case pembrolizumab, on this particular model, what you can see here is, as compared with the vehicle control or the isotype control in the background there for gray, you see an induction or a restoration of immune response in both the stromal and the vascular model when treated with pembrolizumab. We see this by the secretion of a variety of cytokines and other biomarkers. So this shows that the anti-PD-1 drugs do not in fact activate the immune system, they restore its ability to respond in the presence of tumor cells. Now, this particular evaluation, this goes back and looks at diversity plus. So we're looking here to look at the much broader biology outside of the tumor microenvironment. And you can see, for the most part, there is a fairly quiet profile here when we look at either nivolumab or pembrolizumab. And this reflects exactly what is seen in a clinical setting. The PD-1, PD-L1 checkpoint inhibitors can restore immune function within the tumor, and so there is a localized increase in cytokine levels in the tumor, but it is not systemic. And this models exactly what is seen in a clinical scenario, and it's why these drugs are not systemically toxic. If we look at this, again, this is just showing a few other drugs, durvalumab here, nivolumab, and pembrolizumab. We're seeing this restoration of immune response to the tumor cells. And these are classic immuno-oncology agents, so we would expect this. But when we look at three other drugs, erlotinib, gemcitabine, and paclitaxel, these were not designed as immuno-oncology agents. Erlotinib is an EGF receptor tyrosine kinase inhibitor, gemcitabine is a nucleoside analog, and paclitaxel is a microtubule disruptor. What we do see here, though, in this same set of models, is some degree of immune response restoration. So what this suggests then is that these drugs also have an immuno-oncology component to their profile. And in actual fact, this is probably partly what contributes to their efficacy within the clinic, outside of their targeted activity at their various design targets. So we can use this platform not only for immuno-oncology drugs, but for other drugs to show where there may be an additional benefit that was not necessarily designed as part of that drug's profile. Now, if we look at the profile that one might generate from Diversity Plus, that phenotypic profile, the fingerprint that I mentioned, is there anything we can do with that to identify potential toxicities? And can we do that before clinical evaluation and thus save attrition? At the moment, in the US at least, it takes 10 to 15 years typically and approximately $1 billion to develop a new drug. But many of them still fail in the clinic. 90% is a huge failure rate. And of that 90%, about a third of them fail due to unmanageable toxicity, side effects, or adverse events that were not really predicted from some of the evaluations that were conducted as part of a standard IND package. That's not to say those assays are no good. They're perfectly good assays, but they're very targeted, and they don't look at the broader biology together. So the question then is, can we do something in vitro using cellular assays to better model the effect of drugs and whether they do have a potential toxicity issue? So coming back to Biomap here then, we talked a little bit about how we can generate molecular phenotypic profiles, and we can see whether there are common aspects of those profiles with what we have in the database. Now, because we've profiled a very large number of drugs, many of which have known toxicities, we've been able to identify 9 different toxicity signatures. Those toxicity signatures are all different, and they are shown here with different reference drugs that have those known toxicities, along with the pathways that are in part responsible for those toxicities. So if we go to a Biomap Diversity Plus profile and we ask the question, does this drug have a potential toxicity signature? I'll now show you a couple of case studies. This example looks at erlotinib, an ATP-competitive EGF receptor tyrosine kinase inhibitor, and you can see here there are a number of different markers that are modulated outside of the vehicle control. What you will see, though, are 5 different systems with the grey arrows at the bottom in which anti-proliferation is seen. When we take this profile and we match it against the database to look for toxicity signatures, we find 2. We find immunosuppression at 10 micromolar, and we find organ toxicity at concentrations greater than or equal to 3 micromolar. Now, the anti-proliferation that you see in the 5 different systems there in the grey arrows, that probably suggests that organ toxicity may well be a consequence of impaired vascular proliferation. In fact, 2 or 3 of those different systems there model aspect of the vascular biology. So this is probably why organ toxicity is predicted. We also see impaired T-cell function. And again, this is probably what is causing the immunosuppression in this particular case. Not a mechanistic suppression, but a suppression of T-cell expansion. People often ask, well, with erlotinib, one of the key side effects that's seen is an acneiform rash. This assay doesn't really model things like that, but when you think about what might be happening there, the acneiform rash is very likely due to the inhibition of proliferation of hair follicle cells. They are very sensitive to a lot of chemo agents. And when they are inhibited, they have a habit of dying, and then that sets up a secondary inflammation response, which ultimately results in a pruritic acneiform rash. And the second and last example I'm going to give here is of a drug that Pfizer was developing as a CNS agent, and we can talk about this because this has been published. They were looking at an mGlu5 negative allosteric modulator, and it was put forward into GLP toxicity studies. Interestingly, no effects were seen in rats, but in the 14-day and the 90-day non-human primate or monkey studies, type 4 skin hypersensitivity was seen. So, of course, this caused a halt in the evaluation of this drug, and the question then was, what is causing this unpredicted skin hypersensitivity? Can we explain it using in vitro models? And can we devise a path forward for that program using the observations that we make? So this is the Diversity Plus profile for this particular drug here. And you see, as might be expected, it shows a relatively clean profile in many of the systems. But you'll notice in the BT system, a little bit in the SAG system, and also in the LPS system, there are some quite notable responses. In particular, the induction of IL-2 and IL-6 in the BT system, and prostaglandin E2 in the LPS system. So when we take this profile and we look in our database, we do a toxicity signature analysis, we only find one toxicity that relates to this, and that is the prediction of skin irritation at all concentrations tested in this study. And that mirrors exactly what was seen in the non-human primate study. So what we've seen here, we're making the assumption that the monkey response is going to be very similar to a human response. What this suggests then is if this drug were to have been evaluated in humans, it would also cause severe skin irritation, and it's likely due to that induction of IL-2, IL-6, and prostaglandin E2. So then what one can potentially do using this is put in place simple assays to measure the ability of a drug to induce IL-2 or IL-6 from, say, PBMCs, or to induce prostaglandin E2 in a macrophage-type system. One can then use that in an SAR campaign to help remove those undesirable properties from the molecule's phenotype. If then one were to do that successfully and then retest that drug in Diversity Plus here, you would expect to see a disappearance of that induction of IL-2, IL-6, and prostaglandin E2, and thus a disappearance of the skin irritation toxicity signature. So to summarize then, we have a number of in vitro phenotypic cellular assays that can be used to generate very valuable information that help us better understand how a drug is working at the mechanistic level, at the cellular functional level, at the efficacy level, and for predicting genetic biomarkers for drug response, as well as potential toxicity profiles. We can combine various efficacy readouts in looking at human tumor cell line drug responses, and with that genomic database, identify those predictive biomarkers that can then be used to stratify patient populations for a clinical trial, as well as identify potential resistance mechanisms. This can help streamline the trials, which can then obviously speed up the progress, reduce cost, but most importantly, provide hope and promise for patients enrolled in that trial. We can use more complex in vitro co-culture systems then to really look in more detail at how a drug is working. We showed examples where non-immuno-oncology drugs do have an immuno-oncology property to their activity, but we can also use this to look at more translatable findings, things that we see in the clinic that are mirrored in the clinic. And from that, we can use, again, database mining to identify potential toxicity signatures long before clinical candidate selection. So something can then be done about that to help reduce the risk of a drug failing in the clinic and thus reduce drug attrition. So with that, I would like to thank you all very much for your attention, and I would be happy to answer any questions that you may have.